Okay, I'm Carson Wagner, Rick Wagner at the University of Minnesota, and I'm a professor there and Chief Scientific Officer of Tychon. How many people here have had a family member or someone that they know have cancer? Just about everybody. Actually, in the last 2 years, I've had 6 friends get cancer. 3 of them die of cancer. And so one of our goals is to provide and look for life-extending, versatile, and safe cancer immunotherapies. Our company was founded about a year ago. I'm a co-founder along with Inventure and the University of Minnesota. Our CEO is Danny Kunigan, who is an Inventure managing partner. We have strong IP. We have 4 issued patents, actually 1 just issued this week. And we have a pre-Series A funding of about $1.5 million. Now, the pillars of cancer treatment, as you well know, are radiation, surgery, chemotherapy, and most importantly, in the last 5 years, the most excitement has been in immunotherapy, mainly because it can potentially be curative. Our approach is based on a nanotechnology platform called chemical self-assembling nanoparticles, and we can take proteins. There are proteins. We can take a protein, in which case these 2 proteins, one which can target a T cell, one which can target an antigen. We then assemble those into nanorings, which are extremely stable. And they have 2 functionalities, being able to bind to the T cell. And when they bind to the T cell, they bind to the T cell almost irreversibly. These allow us to then target to the cancer cell, activate the cancer cell, and kill the cancer cell. This approach has been validated in the lab, in the test tube, and also in an animal model. We can also have an approach. One of the main things about this is our ability to take a small molecule trimethoprim, an FDA-approved drug, and actually wipe these from the surface. This allows us a critical safety mechanism for allowing us to deactivate the T cells, which is one of the big issues with any T cell-based therapy. Now, advantages, we refer to these as prosthetic antigen receptors, or PARs. They're stable on the cell membrane, and you can modify a T cell membrane in minutes. You don't need genetic engineering. There's no potential long-term toxicities. There are favorable pharmacokinetics and tissue distribution. We have multiple potential antigenic targets we can target. We can be used for blood or solid tumors. And there's a built-in safety mechanism for these. In addition, we have initiated and shown proof of concept that we can be able to use these for image-guided immunotherapy, which would be really nice. The market for this, there is currently 1 bispecific antibody on the market from Amgen. This year alone, it will do $120 million in sales. It's its 2nd year. It had a 50% increase. There's a number of checkpoint inhibitor. This is an antibody as well, which activates the immune system. And this is about $1 billion last year. And the projected market for 2020 is cancer immunotherapy will be an $80 billion market. Right now, we're looking for engaging strategic investors and clinical partners, and thank you.
What's the regulatory path and the time and the capital?
Yeah, so the point for the regulatory path is, of course, you have to file IND and all that, but in this case, it would be treated as a biologic and not a cell-based therapy, which is an advantage. Right now, for example, for CAR T-cell therapies, there really is no understood path for that right now. There's no FDA-approved approach. Even for the production of the materials, there's no FDA-approved pathway for that, and it's having to be worked out. And with this approach, we believe we have an advantage because it's essentially a biotech drug, a chemically induced biotech drug, and so it has that advantage. Capital, we think we need at least probably $10 million for the Series A. The Series A and probably up to $30 million to get to the Series B.
Does the nanoring, it's still comprised of potentially immunogenic?
Yeah, so that's a issue in any immunotherapy. It doesn't even, actually, even if they're humanized, is immunogenicity. You can go to meetings on this, immunogenicity. With your protein. In our hands, when we've done mice experiments, we find actually almost no immunogenicity from these rings. The reason we think for that is because they're kind of unique in that the rings are very stable, and when they're taken up in the cells, they're not processed by the MHCs. They're just held extremely stable, and we know they're stable with the cells. But that's as far as we go. Yes?
In the academic clinical trials anticipated?
Yeah. We would like to So that's the next goal, is to try to, is why we would like to get a preferred clinical partner, to begin having those discussions and begin planning that clinical, the clinical development as soon as possible, actually. So, thank you.